Okay, we're going to do a few annotations on this diagram, in order to talk about the answers to the animal development questions. So, question 1 asked how many cells is a zygote. A zygote is one cell, and that's created when egg and sperm combine. So, we're going to select a color, and I want to color in, let's make that a little bit bigger. We're going to color in the zygote, and that is the one cell. All right, question 2 asked you to describe a blastula. Well, a blastula is just a hollow ball of cells. So one cell divides to become 2, 2 become 4, 4 become 8. About 16, it starts to form this hollow ball of cells that we call a blastula. Now the thing is, every single cell that was formed from that blastula is identical. It's a clone of the zygote. So we're going to color all of these the same color because they're just clones of the original cell. There's been no cell differentiation left. They're just dividing and copying. This next panel shows a cross section of a blastula. So basically we have cut it in half and we can see the inside. So I'm going to color it the same color. And we can see that this is the inside, the hollow ball. And the reason the cells in where I've drawn the arrow are smaller is to show that we're trying to show a three-dimensional structure on a flat piece of paper, which is difficult to do, but they're using some, drawing techniques to show you that those are supposed to be further away than the cells where we did the cross section. All right, question number 3. What begins to happen is how's the blastopore formed? Well, the cells will begin to move inward and they begin to collapse. And it's the collapsing of the cells as those cells collapse inward that forms the blastopore. The blastopore is just the opening into the center of the blastula that's eventually going to become the digestive tract. So these cells here are the ones that are going to start to move inward. And so now we're starting to see some cell differentiation. So the rest of the cells are still original copies from the zygote, but these cells that start to collapse are going to turn into a different type of cell. They're going to become a different part of the gastrula. The blastopore is just the opening into the gastrula. And like I said, that's going to become the digestive system. So where that blastopore forms, depending on where the cells are, they become different structures in our body. So for instance, these outer cells in this layer here become the ectoderm. The ectoderm are the ancestor cells of your skin and basically everything on the outside of an animal. The cells that line the blastopore, the original ones that were collapsing inward, become the endoderm. These are the ancestor cells of the digestive tract and all those other things. So they're basically the inside. And then in the middle there is between those is where the mesoderm forms. And the mesoderm is all the muscles and stuff, everything that's found basically in the middle of an organism. And so scientists refer to these as the embryonic germ or tissue layers. So these are the embryonic ancestor cells of all the cells that you have today, and in the location of where they were in the original embryo. And then finally, the last question was what determines which cells are going to become endo, ecto, or mesoderm. And it's basically the fact that these layers are formed based on their relative position to the blastopore. So that blastopore determines which cells in relation to where it is become endo, ecto, and mesoderm.